Hello. This lesson is defining neurons and glia.
In this lesson, we'll look at the Lego pieces of the nervous system, neurons and glia. We'll start with the Neuron Doctrine before touching on important features, neuroanatomy, and how we classify neurons. Then we'll switch gears to look at glia, the support system of neurons. So, the overarching question I want to answer in this video are, what is a neuron, and what are glia? The Neuron Doctrine gives us the basis for quantifying the principal elements of the nervous system. The history of this theory is fascinating and largely attributed to Santiago Ramon y Cajal, but has been adapted over time to accommodate new discoveries. The Neuron Doctrine states that the brain, and the nervous system for that matter, is composed of neurons, which are distinct, individual cells that communicate via contact. While the Neuron Doctrine does not account for existence of things like electrical synapses, for example, it is a solid model to build the base off of. Perhaps the most important part of the Neuron Doctrine is the communication part, with molecules called neurotransmitters playing a big role in the activity of neurons. For the purposes of this series, the neuron will be defined as an electrically excitable cell capable of exciting other cells. Now that we've got a definition of what kind of cell a neuron is, let's take a look at the components of the neuron. The main body of the cell is called the soma. It's from the Latin word for body, while following the first rule of neuroscience. Always be pretentious. You'll find the nucleus and other organelles like the ER and Golgi body here. The soma has several projections called neurites, but one of them is quite special. This special guy is the axon, which arises from an axon hillock, conducting electricity away from the soma. The axon can be quite long and even branch out with more than one target cell. And it's not just electricity that runs down the axons. Proteins and neurotransmitters also need to make their way through, and this can be done in a fast or slow manner. Slow transport is just simple diffusion, so the organelles in the soma will just pump out a bunch of their product, and some of it will float down at its own pace. Fast transport, on the other hand, uses motor proteins to ferry items around using the microtubules. Think of the motor proteins as trucks carrying cargo down a microtubule highway. In this analogy, the gas in our trucks is our good friend ATP. In the axon, cargo moving away from the soma is carried by a motor protein called kinesin, while cargo moving towards the soma is carried by a protein called dynein. So why are we moving stuff down the axon anyway? What's at the end of it? Here, we find the axon terminal, where all the presynaptic machinery lives. The space between one neuron and the next is called the synaptic cleft, and anything that's needed to put stuff into the cleft lives here at the axon terminal. If we were to continue our trucking metaphor from before, we could compare the axon terminal to a local distribution center, where some final modifications and packaging can occur before shipping across the synapse. This will of course take energy, so the axon terminal is rich in mitochondria to generate the ATP needed for this process to occur, as well as vesicles, which bag up products for transport into the synaptic cleft, kind of like the cardboard boxes your online deliveries come in. Across from the synaptic cleft, we don't see another soma, but dendrites that make up the postsynaptic structures. Dendrites will receive and integrate signals from other neurons and are really spiny. Unlike axons, they're rich in ribosomes, which can produce proteins as a response to synaptic transmission. We can group the axon terminal, cleft, and dendrites together into a single structure simply called the synapse. We just hit the basic anatomy of the average neuron, or your Platonic ideal of a neuron, if you will. But there's a huge amount of diversity in how these structures manifest. For example, the neurons involved in making sure you hear what I'm saying are different from the neurons in processing what I'm saying, are different from the neuron reminding you to blink right about now. As such, we can put neurons into a whole bunch of different buckets, depending on what we're personally interested in. Why don't we look at a couple strategies to do just that? The first way we can classify neurons is by shape, which in practice is based on the number of neurites. There's unipolar, bipolar, and multipolar. Unipolar neurons are usually found in invertebrates, but there is some nuance here with pseudo-unipolar neurons where a single projection branches out later. Bipolar and multipolar cells are pretty self-explanatory, having two or more than two projections respectively. We'll see bipolar neurons acting like connecting wires in our sensory areas, like in vision. Multipolar cells are seen all around the body. Just look at your muscles. Another way to break up neurons is based on what they release. You might have heard of dopamine or adrenaline as neurotransmitters before. Well, a neuron that releases dopamine can be called dopaminergic, and a neuron that releases adrenaline can be called adrenergic. Similarly, we can get cholinergic, serotonergic, etc., etc. It goes on. Another grouping we can look at is based on the firing patterns, such as bursting versus continuous, but that'll make a lot more sense once we use it in the context of communication, so I'll hold off on the nitty-gritty for now. Finally, there's some special cases where a neuron is named after something or someone, as is the case of Purkinje cells and Renshaw cells. All in all, there's a whole host of ways to classify neurons, and it can be helpful to keep in mind when we're studying them. Neurons are not the only thing that make up a nervous system. In fact, only about a quarter of the cells in the brain are actual neurons. The other portion, the majority, is made up by glia, which offer a range of supporting roles to the neurons, whether it's by filling space between neurons, repairing things, working on immune surveillance, or regulating the environment around the neuron. You name it, glia will probably do it. While neurons are still the star of the show, we don't want to completely forget about the supporting cast, right? So let's take a quick tour into a few prominent types of glia. Astrocytes are quite abundant and have a lot more subtypes. They've got a lot of roles, some that we don't even know yet. But the gist of it is that astrocytes provide nutrients and regulate the environment by helping clean up the synapse after transmission. Oligodendrocytes and Schwann cells, on the other hand, have a single function, forming the myelin sheath on the axon. The myelin sheath is a structure that makes the movement of electricity much more efficient and is on most neurons. Oligodendrocytes are found in the brain and spinal cord, the central nervous system, while Schwann cells wrap around axons in the peripheral nervous system. Finally, microglia help remove debris from dead and degenerated cells, and help with the immune function by destroying infectious organisms. Other functions are kind of poorly understood. Okay, we got through a lot today, so pat yourself on the back. We covered what a neuron was, what its parts are, and how to classify them, and a couple types of glia. I left timestamps in the description below if you wanted to go back to a specific section. Thank you for watching, and I hope to see you next time.